Welcome back. So, this is another episode of Journal Club. If it's your first time to the podcast, welcome. STEMdentity tends to focus more on episodes around people's career journeys or specific topics that I have found myself and others really struggling with in science and engineering type careers. But also because I like to have fun and learn new things, I do Journal Club once a month where I do a bit of a dive. Usually I go to sites like Science Daily and I see what sparks my interest. And it's been a really great experience. I've gotten to meet people I would never get to meet otherwise. I've gotten to read papers I never would have read otherwise. So I'm super grateful that scientists and engineers are willing to hop on with a complete stranger and talk about their work. So my guest today is Dr. Audrey Lin. Audrey just wrapped up a postdoc at the American Museum of Natural History, and we're talking about her Proceedings of the National Academy of Science, or PNAS, paper, A Legacy of Genetic Entanglement with Wolves Shapes Modern Dogs. And I saw this article and I got excited because I know I talk about my cats a lot. I got two kittens. I got kitten brain all the time. But we also have two dogs. They're awesome. One is a German Shepherd Golden Retriever mix, and the other is some sort of hardy mixed breed, right? Big dogs pull me around. We have fun, take them outside, do all the good things. So Audrey is such an amazing scientist. She's so excited about her work. She does really cool things. Her paper is really cool. We dive into some other papers that she's done as well a little bit. Very excited about it. Let's dig in. One of my most favorite ways of getting excited about science and engineering is talking to scientists and engineers who are really excited about what they do. Welcome to STEMdentity Journal Club. Every month, we're going to take an article and talk to its lead author to learn more about the amazing work that they've done. Let's meet today's guest. Awesome. Dr. Audrey Lin, I'm so excited you're here today with me, and I know you're incredibly busy. You're moving to Vienna soon, which we'll talk about near the end. So I really appreciate you taking a little time out of your day to talk about your recent paper in PNAS, The Legacy or A Legacy of Genetic Entanglement with Wolves Shapes Modern Dogs. Before we dive into the paper, I'd really like to hear a little bit more about yourself. I see that right now you're at the American Museum of Natural History in New York. Before that, I saw you were at the Smithsonian, which, that's a name that pretty much everyone recognizes immediately. You just wrapped up a scholarship. You went to Oxford. You did a ton of things. Can you give, I mean, this is a big sprawling question, but can you give kind of a summary of like, how did you choose your research path? What were the things along the way that really inspired you to get to where you are today?
Sure. So if somebody had told me that this is where I was going to be, and this is what I was going to end up studying, like when I was a kid, I would not have believed it. I really only became interested in science much later on. I've always thought that I was going to be like a writer or like a historian or a librarian or something like that. I was a very indoors oriented kid, and was just like watching TV and reading books all the time. So I came into science like relatively indirectly through mainly pop culture depictions
Really?
of scientists. The Scully effect. How Dana Scully from the X-Files had shown girls that it's possible to be a scientist, and had presented herself in a really relatable, admirable, badass way. That was like that had drawn me more into science than anything like in school, to be honest, because I never imagined that being a scientist could be anything like that. Although, like, we're not hunting aliens, but um, I had gone back to school in higher education later, like in between graduating high school and going to college. There was about a 5-year gap because I didn't know what to do. So I was working for like various jobs, customer service, retail, that sort of thing. And I had realized this was so one thing I like to ask as sort of an icebreaker when I'm talking to like a group of students in science is what's your science origin story, right?
Mm.
Like what event or events had uh put you on the path to being the science superhero, so to speak. And I would say for me, I was in my cubicle job in Southern California, and I was studying for my driver's license.
Mm.
And they have the driver's license manual, the driver manual that you have to study to pass the test and everything. And I realized that I was enjoying the studying aspect a little too much. Like I knew that I shouldn't be enjoying this. So it was at that point in which I finally was like, okay, I think this means that I should probably go to school. Um, so I had started at a community college in Las Vegas, actually, and had transferred my credits to the University of Nevada, Las Vegas. And I got my undergrad there. And also another thing to answer your question on I think you mentioned something about like advice or something like that.
Yeah.
I would have to say I would also not be where I am if I was very stubborn in following a specific path, you
Mm.
know?
Hmm.
I had originally planned on getting a degree in clinical laboratory sciences, and when I was one course credit away from getting into the program, the university had cut the program completely. And so I had to pivot. And it actually turned out really well for me because I was able to take other classes in biology and in anthropology that I would not have been able to take if I had gotten into that clinical laboratory sciences program, which is more of an occupational training sort of degree program. I was able to take, microbial ecology and molecular biology and forensic anthropology and evolutionary medicine, and it just completely blew my mind, the sorts of research that people were doing. I was an undergrad in the late 2000s, the early teens, 2010s, and this was when the microbiome research was really picking up, and I thought that it was just so incredible and a kind of a paradigm shifting worldview of even in how we regard ourselves, like not just human, but a amalgamation of like millions, billions, trillions of other organisms like living together. So then I wanted to see if I could go further, and so I decided to go to grad school and wanted to see how much further I can I can go. And so I just like continued and continued. And when I started at Oxford, was actually originally working on viruses and cancer, but it wasn't a good fit with my first supervisor, so
Mm.
I had ended up switching supervisors and switching projects completely, and so that was how I had ended up working on ancient DNA and paleogenomics on animals. And again, like even when I had started like grad school, I never imagined that this is what I was going to do. I didn't care about, like the genomes, the evolutionary history of pigs and chickens, but now it's like, wow, it's so cool. So yeah, be open-minded about possibilities and yeah. So that was long and meandering. I hope I answered your question.
No, you definitely did. That was amazing. There were so many cool little bits out of that. Like, I love the science origin superhero story. I was kind of thinking about mine when you were talking, and I feel like mine was maybe a little less inspiring. I was sort of strongly nudged by young, or when I was young, I was strongly nudged by teachers around me. Like they could see that I was pretty intelligent and pretty just like bored
Mhm.
with whatever was going on in class. So they kept like being like, here's this, Carl Sagan, Cosmos, go read this in the corner or something. And it wasn't like I loved it, but I also liked that acknowledgement and recognition. So I was like, well, I guess this is
Mhm.
what I'm supposed to do. So it took a little while. It wasn't really until undergrad when I was in a physics class, my first physics class ever, that
Mhm.
the teachers were actually having fun. I was like, oh, science could be fun. It's not just what you're supposed to do because, you were told by someone else to go do it. So, yeah,
Yeah.
my origin story is maybe a little more smeared out and less inspiring, but I love that. I love the Dana Scully. I watched that as an adult, not as a kid.
Mhm.
And I was like, oh, this woman is really cool. But also, I didn't see myself in the lab, so I didn't connect myself to her. I've always been kind of more of a computery type person, computer
Mhm.
research type person. And I also think it's really cool that, I think a lot of us, when we think about Oxford and going to grad school for Oxford, we kind of assume it's either a privileged class of person who was born into a certain type of family and, they've just been able to go that path their entire lives or stories like that. So to hear that you took a break after high school, you didn't just go straight into college and then grad school and everything, that you can not know exactly what you want to do and still end up doing wonderful things almost by not accident, but not, you hadn't planned when you were six
Mhm.
that you were going to go to Oxford for ancient DNA research. You were like, I like history. But now you're kind of doing history with ancient
Yeah.
DNA research in a way, it seems like.
Yeah, exactly. Like it's really cool now to be in this position and sort of reflecting on on what my interests were when I was younger and coming to the realization that there is still like continuity there even if I don't didn't realize it at the time. Like for example, with all of the dog related research that I'm doing now, I literally it was like about a month ago when I had remembered when I was 9, 10, 11 years old, I was obsessed with dogs. Like, you know how there's the cliche of like little girls being obsessed with horses.
Yeah.
It was that with me and dogs and like my favorite. I had all of these those eyewitness encyclopedia of dog breeds books there's like several of them from different companies. I had a whole bunch of them and I would spend so much time like pouring through those books and comparing and contrasting what they would say in between them and sort of synthesizing the information. And I'm like, oh, I was doing this at like 10 and I was like giving school presentation about pugs, you know. Yeah, I encourage everybody to sort of think about these sorts of things.
Yeah.
It's fun.
Yeah, and do you feel like that? I love that because yeah, as a kid, I did the what was it? It was like the flow charts. Like, here's this rock,
Hmm.
and follow the steps to. And now,
Yeah.
I pull out Merlin Bird ID all the time, and I'm just like, I always want to do the follow the steps because it just like it scratches that itch of like this, not that, this, not that. I
Yeah.
love that kind of deductive reasoning. Do you feel like it was helpful for you to have that 5 year gap between, high school and college to give yourself a little, I don't know, grounding or something before you went back to school?
Oh yes, definitely. I mean, high school was not a good time for me. I was not a good student. Um, and I mean there were classes that there are some classes that I liked, but I was also at the time much more interested in like movies and TV shows and going to concerts and in the late 90s, early 2000s, it was a great time for music if you were living in Southern California. So so it was really good to sort of get out and get more life experience and having to interact with with like people daily and also learn have jobs in which you realize, like, I really don't want to do this, like it's really important. I
Yeah,
mean, yeah, yeah.
I have a whole philosophy around that where it's like you should just try things and figure out what you do and don't like because probably there were a couple things you liked about each of those crappy jobs that you had to do,
Mhm.
but that wasn't going to be your forever life, and that's good to, acknowledge and move on and find something that fits you better. So after grad school, you went to the Smithsonian. How'd that come about?
Yeah. So I had applied for several postdoc fellowships, and this was the only fellowship uh, in which I got an offer on. So,
I mean, go to the
yeah.
top, that's fine.
Yeah, exactly. And the thing was, like, I would have never applied if I hadn't gone to a conference, an ancient DNA conference, and was hanging around. At conferences, there's the presenting research and learning about new research aspect of it. But what is arguably even more important is the networking
Mhm.
and like what happens during the coffee meetings and also at like the bar afterwards. And I knew this, right? So I went to a conference. It was about 5 months before I was set to defend my thesis. And I still didn't have anything lined up for afterwards. So I was really determined to make connections with PIs, group leaders for a postdoc. And so that was what I did. And I was sitting at a table, talking to a postdoc, and this was in Jena in Germany,
Mm.
right? Which is a very, I would say, historically and culturally significant place for German Romanticism,
Mm.
and for philosophy, for science, Sorrows of what is it, Young Werther by Goethe, like all was birthed there. So I was talking about this to the postdoc, and then all of a sudden, because I was at the same table with my supervisor, he called me over because he was with two group leaders who were based at the Smithsonian, and he hyped me up like he had been apparently hyping me up in talking to him, and then he called me over and made the introduction, and so I was able to. And we made plans to write up what is it, a research proposal for the Smithsonian postdoc fellowships together with both of them.
Mm.
So that was what I did. And like the thing is, so I got my minor in anthropology, in the US, and I had thought that the Smithsonian was just like the highest of the high. I didn't think that it was possible or achievable. It never even occurred to me that I could do anything there, which is absurd because like I was doing a PhD at Oxford, and yeah, that had brought me down to earth in a good way in a bit. Um, so so yes, that was how I had learned about this fellowship there, and that it was possible to apply, and that I was considered like a strong candidate and
Mhm.
all of that. And so I wrote up. I actually applied to two different fellowships with the same project, and I had ended up getting both of them. I had gotten, and the thing was, like, I had come up with about like 95% of the like I wrote about 95% of the research proposal. Like everything, everything was me. And so that it just felt so incredibly validating. Since during your PhD, I mean, you're doing the work, and you are synthesizing your results, but and contextualizing it, but it is also led by your advisors, you
Mhm.
know? The project ideas themselves, or they fit within the umbrella of, you know, like your advisor's research aims, like within his group. So, but in the case of this research proposal, it was all me. So yeah, that was probably the first time I had ever felt like that level of validation for what I can do, what I'm all about, and people acknowledging that. Um, I think a common theme now that in my sort of stream of consciousness rambling is there's a lot of second guessing, you know, in this journey. It's something I think most of us struggle with,
Mhm.
the imposter syndrome thing, which makes it even more poignant to have these sorts of moments that yank you out of that.
Yeah, that's really beautiful. I love that. I think it's a big jump and not everyone makes it and not everyone wants to make it, which is absolutely fine, of going from kind of that I'm working for someone, I'm doing their projects. You have some amount of autonomy, right? You're not just being like,
Hmm.
okay, tell me step one, two, three, four,
Oh
one
yeah, yeah, yeah.
500, but right, you're still kind of being guided to actually doing an independent research project and leading your own projects. And I mean, frankly, I'm still figuring out how to do that some days. Some
Hmm.
days I'm kind of like, wait, what do I do? Um, so I think that's really amazing. And I also want to pick up another piece a little bit back, which was that networking piece, how you felt like, okay, I can apply for something at the
Yeah.
Smithsonian because I've talked to people. And I think it's easy to overlook that, especially because a lot of scientists, myself included, are pretty introverted. We don't thrive at conferences. We don't love going up to people we don't know. But I've certainly never gotten a job without knowing the person before, right? In the times of my life where I've been searching for a job, if I cold apply, I've cold applied to maybe, I mean, not a lot of jobs, to be honest.
Hmm.
We'll say 20 to 30. I heard back from one, and it was like a
Yeah.
pre-screening interview, which I totally flopped because I couldn't use the interviews. But yeah, I think that's really key of just, remembering that maybe it isn't your comfort zone to go and network,
Hmm.
but it still can lead to such incredible things down the road that you can't even foresee. Because now you're at the American Museum of Natural History in New York. You were at the Smithsonian for 3, 4 years, give or take,
3 years, yeah.
3 years. And your current project. So I saw, when I was looking at your profiles, that you have this Gerst, I'm going to pronounce it wrong, Gerstner
Yeah, the Gerstner. Yes.
scholarship. And that, the profile page for that scholarship, so that you were going to be working on genetic roots of I can't even say the word
Virulent.
virulence, thank you,
Virulent,
in
yeah.
historical
It's all right.
viral diseases, working with bats.
Hmm.
And then the paper we're talking about today, which we are now kind of moving into is about dogs and wolves. So, had this paper that you did in PNAS, which is no small journal, right? It's one of the big three, PNAS, Nature, and Science.
Mhm.
How did how did this current project on dogs and wolves come about? Was it a pivot from your original plans with the scholarship, or have you been kind of doing both at the same time?
So I had been doing both, and this was actually sort of a continuation of the research that I was doing at the Smithsonian. So I was originally planning on doing the more virus related projects at the Smithsonian. I had moved there in January 2020 and was able to have enough time for just for to try to get things into place, and then the pandemic shutdowns happened, and then I wasn't able to do anything, as in we weren't allowed to go to the collections, we weren't allowed to access the labs, we weren't allowed to, and this was this was a data generation project. Like I had to go, I was planning on accessing collections material which is birds, waterfowl, ducks and swans, and so forth, that had been treated with formaldehyde and preserved in jars of ethanol. And these birds were more than 100 years old, 150 years old, and sampling from them and trying to recover fragments of viral RNA. Influenza RNA was what I was specifically interested in. But I couldn't do that with everything shut down. I so the only things that I could do was work with public data.
Hmm.
And so the dog project, this current one that you asked about, it had its origins in that I was interested in sort of exploring the evolution of different canids by constructing large trees from their genomes and seeing like where the placements where they were situated, all the different wolves, the different wolf subspecies, coyotes, um, uh, dogs, and also like more extant canids like the jackals and so forth. And then it just sort of like sat for a while. Like I made the trees and it didn't go any further than that. I had also then had started working on like a different dog project, which you may have read about when you were reading up on me on the Coast Salish woolly dog.
Right. Which was
Yeah.
published in Science. Amazing. Congratulations.
Thank you.
Yeah.
Yeah, with that project, there was we did utilize a lot of public data, but it hinged on getting DNA from this one woolly dog specimen, and so it was much easier to, like, once restrictions had lifted a bit, it was easier to go in and sample and do the lab work from one specimen versus what my original project had required, which was sampling from like 50 to 100
Hmm.
like specimens. So yeah, like all, all of my dog projects actually have been sort of side projects that became primary.
Yeah.
And so I'm like really grateful for that, but it also emphasizes the importance of flexibility.
Mm-hmm. Yeah. And I mean, there's still, I feel like the tie is maybe still the ancient DNA, but then, yeah, sort of moving from
Yeah.
birds to dogs, fairly different. As a side question, what is a woolly dog?
Oh, so the woolly dog was a specific type of dog that the Coast Salish or the Coast Salish and other peoples on the Pacific Northwest coast had maintained they bred for hundreds up to perhaps thousands of years prior to European contact. They have all of these origin stories involving these dogs. There are many different communities that have different stories
Hmm.
about these dogs, and they had raised these dogs for their hair. So the Coast Salish and the other peoples who are not Coast Salish along the same region, they are weaving people.
Hmm.
So they weave incredibly intricate, basketry from cedar bark and also textiles like blankets, highly significant blankets. And a lot of these communities, they didn't have access to mountain goat, the wool.
Mhm.
And also there was no sheep there, but what they did have are dogs.
Huh.
So they had selectively bred these dogs to have woolly type hair
Okay.
that they were collecting, and it was only the high-ranking women in communities who were allowed to keep these dogs, and all of the knowledge about raising the and keeping the dogs. It was generational knowledge that was passed on between the women in successive generations. And so these dogs are now considered extinct, I guess, or at least the practice of keeping the woolly dogs. Was already starting to go out in the second half to the third quarter of the 19th century, and there were stragglers into the 20th century, but they don't exist anymore. And it was from colonial pressures, forced assimilation that had forced the Coast Salish peoples to give up these dogs. So yes, so
Yeah.
that's what woolly dogs are.
Cool. Yeah, so for your PNAS paper,
Hmm.
there's almost 2,700 samples. And I believe that's a mixture, right, of
Yeah.
and dogs. So was this all public data? You kind of alluded, but maybe let's talk about
Yeah.
the data set a little bit more. Because the listeners might not know a lot about genetic sampling. How were these samples collected over time? Was it full DNA lists? That's not the right term, but yeah, you
Hmm.
can you can enlighten us on what it was.
All right, yeah. So we used all public data. We used a combination of, so these are all huge, huge files, like computer files that can be up to, I don't know, like 300 gigabytes of data per, like, for one file. That one genome. And I was just downloading like everything that I could find that was marked on the public repositories like GeneBank, the European Nash European Nucleotide Archive (ENA). So GeneBank is a public repository of biological data, of genetic data, of protein data, of RNA data that is maintained by NCBI, well, by the government, basically, right? By NIH. It's part of NIH. And so so this database is like one of think uh, uh I think two or three internationally in which it's like a public good sort of thing, right? So the Europeans have the ENA, and the data is often mirrored between the different databases, and I think Japan has one too. So yeah, so there's the raw so sometimes it's raw genetic data, like it's just all like the sequencing data. Sometimes the data has been processed in a way to have perhaps all of the non-host sequencing reads taken out. Like for
Okay.
example, if you are like for an ancient DNA sample, you of say taken from a dog tooth, right? You're just interested in the dog.
Mhm.
So, but like the vast majority of what you're going to sequence via this method called shotgun sequencing, in which you're sequencing everything that's in the sample, like the vast majority is going to be environmental, or microbial, contaminants. It's going to come from like human skin. It's bacteria, it's fungi, it's whatever. So if you look at an ancient DNA paper and they talk about endogenous content,
Mm.
this is what they're referring to. Like the endogenous is content of 2%. So within that sample, 2% is dog.
Yeah.
That's yeah. And with ancient DNA, with the DNA, host DNA, the older it is, the more degraded it is. It's very common for the endogenous content to be like much less than 1%.
Really.
Yeah. And I mean, it's a lot less informative to have to stitch together um, a genome from a sample in which less than 1% is what you're interested in, but we make do with it. So there was some of that in our data set, but there also was a vast amount of modern data. Um, Modern genomes from the Dog 10K project, which is a huge consortium that had been going on for years. I that from many different institutions around the world in which they want to sequence. I think the aim is to sequence like 10,000 like high quality canid genomes and also provide informative metadata, which is incredibly important to have because like it has information about the maybe the sex of the individual, the if it's a breed dog, what type of breed it is, like where the sample came from, what lab produced the data. All of this is really important information to know. So we utilize, and it's an incredible resource. So we utilize all of the information, all of the so much data from the Dog 10K project for for this. And so yeah, that's where all the data came from.
How much of the project was just sort of the data cleaning? Because I'm imagining you're getting all these data from different data
Mhm.
sources. I've worked on atmospheric chemistry projects where it's like 10 different instruments. And of course, every single instrument is completely different in how they report data. So it's months of just cleaning and combining and harmonizing before you can actually do anything cool. Was it, especially when you have 300 gigabyte files,
Yeah.
sort of deal with the magnitude of the data cleaning?
So with so there were some ancient genomes in which we didn't have to. I mean, the cleaning part isn't that bad if
Okay.
like there's pretty good pipelines for this already. The problem is if the genome quality is like not very good. So there we had to. It took a while to as you said, sort of like harmonizing everything and also making sure that the formatting is comparable to each other because all this data was generated from many different lab groups and in which also the sequencing chemistry, sequencing approaches, and the steps leading to that may have differed as well, and there may have also been other chemical treatments of like the samples prior to sequencing that could be reflected in the data that we had to account for that. And also, sometimes the metadata is just wrong, and this would be really obvious if with when we would see like really weird result, for example. So yeah, that certainly does take a bit of time and a lot of CPU crunching.
Yeah, I'm imagining you're not doing this on your friendly home laptop. Did you have access to some sort of supercomputer?
Oh God, yeah. Access to a high performance cluster.
Mm.
Uh, very important. We were using the Smithsonian's HPC.
Great. Yeah, and so the paper itself, I mean, there's so many cool different things you go into. You look at wolf ancestry and breed traits, dog
Mhm.
breed traits, gene flow, olfactory. So for you, you wrote this paper. This is your baby, your work. What do you feel were some of the key findings, some of your favorite findings from this paper?
So I think the key finding is actually just being able to see that evidence of the post-domestication ancestry, like hybridization between dogs and wolves, like in the genomes. I mean, dogs are like they came from gray wolves, right? And they were domesticated from wolves, although not from the wolves that are alive today. They were domesticated from a population of gray wolves that had once existed, what, like 25,000 years ago, that no longer exist anymore. And we don't know
Didn't know that.
where, yeah. We
Interesting.
don't we don't know where like what they look like. We haven't found the smoking gun wolf
Mm.
population that dogs have arisen from. Everything that we know about this population of wolves that were the progenitor of domestic dogs is like inferred from looking at ancient DNA of early dogs and also ancient DNA of wolves and seeing that they're quite different.
Mm.
Um, so um um so wait, I forgot what my original and key findings. There's a lot of like oral and written traditions of hybridization between dogs and wolves. Like even in like antiquity in ancient Rome and ancient Greece, you have a lot of these sorts of stories, but we don't see any evidence of this like in the ancient dog genomes. Like we haven't found anything that would support these stories. And so we thought that maybe this has something to do with the fact that you just can't have very much wolf in dogs. If you want to maintain control over the dog and for like whatever purpose you would have for the dog. The exception to this rule, to this, though, would be like modern breed dogs that people purposefully had hybridized dogs with wolves, like the wolf dogs, Czechoslovakian wolf dogs, Sarlus wolf dogs, and I think that there's like a few others of these wolf dog breeds in the world. So yeah, we had gone all this time, have not been able to really detect any clear evidence of hybridization, of admixture of gene flow from wolves into dogs. Until we had used this very powerful, very sensitive method called local ancestry inference, which is a population genetics approach that was initially used in detecting archaic uh, human ancestry, archaic hominid ancestry in human populations, right? Like detecting Neanderthal ancestry and Denisovan ancestry in human genomes. So this is what we used we decided to use this method on dogs and wolves to see what we could find, and so that it was so finding like very small but detectable post-domestication wolf ancestry in the majority of dog genomes was surprising, and then seeing like sort of broad large-scale patterns in different dog groupings was also surprising, and so yeah, that's what I like about the paper. It's a bit controversial with some of my other dog colleagues, research colleagues, but that's okay.
Hey, science is all about controversy. I mean, I love that you started your discussion out with, citing Aristotle and Pliny talking about dog wolf hybridization. So, like, do we have a basic understanding of how this would work? Because, right, my basic,
Mhm.
going back to high school biology, I'm like, well, you have one wolf parent, one dog parent, and now the child is going to be 50-50. And
Mhm.
now you're seeing, things in sort of the less than a percent to a few percent wolf,
Yeah.
a dog. So is it just that, yeah, maybe back in Pliny's day, they bred wolves and dogs, but now we just have a few percent DNA left. So, how is that sort of trickling down through the generations?
So we can sort of back of the envelope calculate how many generations have, or how long it's been since the wolf had entered the, well, I guess the lineage of the dog. Depending on how large, basically how big the chunks of the wolf DNA are in the genome.
Okay.
So the smaller it is, because with the smaller the chunks of DNA, the older the lineage is. The more far back in time the wolf hybridized. So and this is because with the nuclear genome, there's recombination that goes on between the parent chromosomes, um, and the creation of the offspring.
Mhm.
Like they cross over, they mix with each other, and the pieces of DNA, they come apart, I'm and all of that. I'm like explaining this like middle school level bio high school
Okay, that's
biology
about
very
where I'm
badly,
at
but
biology level. I
yeah.
like air pollution. It's not alive, so
So anyway, there's like so there's physical mechanisms that enable this mixing to happen, and for the chunks of like the DNA to become smaller and smaller over time. So yeah, and then we can sort of calculate, depending on like how big the chunks are, how long it's been, and we were sort of roughly saying that using the generation time of 3 years, and so like a lot of well, so what was it? What it in that paper? Sorry, in
no,
my
you're
paper, was it like up to like 20,000 generations we were able to detect, or something like that?
I'm not sure. Yeah, I don't remember that number, but I'm the same way. I publish something and I immediately forget 80% of what's in the paper. So, but yeah, that's really impressive. I think, another thing that I was really struck by with this paper, maybe this is just I'm reading in the wrong sectors of science, but your graphics are just they're so big, right? I'm used to like, here's my tiny 1/4 page is a very hardy graphic, but no, you've got these beautiful, sprawling, full page, intricate graphics. I'm assuming there's a lot of cool coding packages that are out there for doing all this, but is this something you've seen evolve over time since you've been in science that we've gotten to go to larger graphics, or is this just your field has always understood the value of a good graphic and, can put it out there?
In biology, having pretty figures goes really far. Right? I'm not like very I have colleagues and friends who are really good at this.
Mhm.
And it takes a lot of time to do this. I mean, typically what you're supposed to do is like do the analysis in like R,
Mhm.
or something like that. R will generate the figure for you, right? The
Yeah.
plot, the graph. And ideally, you should have everything coded in R that will generate the as close to complete figure and graph or whatever as possible. Um, this is generally not the case for most people that I know, and also including myself. Having everything coded in art, like, well, the whole point of it is to make it as reproducible as possible, as
Mhm.
quickly as possible. But like it's usually not the case. So we pretty it up in like Adobe Illustrator.
That's totally fair. Yeah, in my field, there's a lot of time series of air pollutants, and
Hmm.
frankly, it's really hard to make a time series of an air pollutant pretty or exciting. It's just,
Yeah.
it's like not that interesting. So I loved the graphics. I don't know if I understand how to read them all very carefully, but I'm sure with, a little bit of training, I could get there. And speaking of graphics, communication in general, right? It's so important. I have this whole
Hmm.
thing about how like we're not really trained to be communicators. We're just expected to be good at communicating.
Hmm.
And certainly, this interview has been amazing. You're a great communicator. This paper has been picked up by different news outlets. I found it first in Science Daily, which I like to go to, inspire
Hmm.
myself outside of my own field. But I'm pretty sure I even saw an Instagram post that quoted your paper because it was something like 0.2% of the Chihuahua genome is,
Hmm.
from wolves or something like that.
Hmm.
And then I read your paper. I'm like, wait, hold on, yeah, that's where that came from. So have you had kind of a experience over time with communication? Has this paper been one of the first times that you already have a science paper out, so maybe you've
Hmm.
you've already kind of done this racket, but like getting to talk to people about what you're doing that aren't experts in your field, how has that gone?
So the previous science paper was had a lot of media attention, so I got a lot of practice from that. The approach to the interviews and telling the story for the public was a bit different in the previous paper because it was a collaboration with my First Nations and tribal, well, my Coast Salish colleagues, and so it was really important that all of the journalists got to talk to them a lot
Mm-hmm.
with the scientists, you
Yeah.
know. So, whereas with this paper, I mean, it's like it's just they're just talking to me and our co-author Logan Kistler. So, and it was all about it. It was just all about science itself.
Hmm.
So. Yeah, yeah, I've talked to quite a few journalists. I mean the communication, I agree with you. It is incredibly important and I think it's a I do my best to spend as much time as necessary to make sure that the journalist who I'm talking to gets understands like all of the points I want, to come across.
Yeah.
Because I mean, it's inevitable that there will be a sort of trickling down and degrading the of the findings of the paper. Like it's sort of like a game of telephone
Hmm.
when it comes to science reporting, especially since they are referencing a paper that references or they're referencing an article that references another public article and so forth.
Hmm.
So you try your best like at the origin to make sure that they have everything right. And like I would have to say that like all of the reporters, the journalists who I've talked to are have been like really good about that. I mean, every like if you're a science journalist, you want to make sure that you're reporting accurately, you know.
And I think that's a
So,
really good tip. Spend the time up front. Is it's
mhm.
tempting for those of us who have really packed schedules. Okay, yeah, I'll talk to this reporter from 2 to 2:30, and then 2:30 I have another call or whatever. But to try to, if possible, just have a little time after your scheduled appointment, just in case it goes over, like our
Yeah.
interview is going over right now. I apologize for being
No, it's fine.
gracious with your time. But yeah, just to tell that story, I think is a great tip. Certainly I've not had a ton of interviews. I won one very large grant back in the Biden administration that has since been canceled, and that was kind of my first time with interviews, and it was very stressful. And I got some good tips, but I think I also just had to kind of learn on the fly. So
Yeah.
hopefully it was good enough. But just like everything else we do, right, there's maybe a little bit of training out there, or we go and talk to the right person who steers us in kind of the right direction, but there's nothing like doing it to just learn how to do it. So,
Exactly.
as I
Yeah.
mentioned, we've already gone over, you've been amazing with your time. I did see that up next for you is going to University of Vienna for
Mhm.
4 years funded. Amazing. Congratulations. Do you know what your
Thank
research is going to
you.
be out there yet?
I am planning on continuing building upon my dog research. I have incredible collaborations with various Coast Salish First Nations in
Mm.
and we are very interested in learning more about the dogs that they had. So trying currently trying to generate some data on that and yeah, like all I'm just looking forward to being able to tell stories about dogs of the past. I
Yeah.
yeah, I just like I find well, I clearly find them just like endlessly interesting and I also find them interesting in that they are reflections of human culture. You
Mm.
know, they were the first they're the first organism that had ever been domesticated by humans. Like way almost probably like more than 10,000 years before any other organism, plant or animal. So they've been with us the longest, and you find and you see, and we have taken them to ev with us to all parts of the world. We have molded them for whatever purpose that we needed them for, whether it's for transportation, for guarding, for for war, for companionship. They are a reflection of cultural needs, of environmental differences, of um, of cultural trends, you know? Like there's so many breed dogs that had once existed that don't exist anymore, you know? Because people just didn't whatever they were bred for specifically, there wasn't any need or desire for them to be kept anymore. So, some of the dogs, probably the majority of dog breeds that exist today might not exist. Like, probably won't exist 100 years from now. So, yeah, there's that. Telling more stories about that is what I'm planning on doing in the next few years.
That's so beautiful. Yeah, I mean, I think you're a wonderful storyteller. I can't wait. I'm definitely adding you to my Google Scholar follow, and I'm hoping, fingers crossed, maybe in a few years I can have you and maybe some of your co-stylish colleagues on for another interview for your cool work out in University of Vienna. I'm a little staggered to think about, yeah, 100 years from now, who knows what dog breeds are going to be out there, right? Because I just feel like, you know, I've had a Staffordshire Bull Terrier. I have a
Mhm.
Golden Retriever, German Shepherd mix right now, and then some other terrier. And yeah, so when they misbehave, I'm going to tell them, watch it. Otherwise, your breed is going to go out of existence. So I'm sure it won't do anything. They're pretty
Yeah.
lucky,
Or I
happy-go-lucky dogs.
Yeah. I think what's most like what is probably likely is that I mean there probably will be like a Staffordshire Bull Terrier, but
Yeah.
they'll look very different. You
Mhm.
know, like English Bulldogs from 150 years ago, they looked very different. They had long legs, and
They-
yeah.
Well, I think
So,
they need to work on softer heads because my Bull Terrier broke my leg once by just
oh
running
my
right into it.
God.
So So yeah, if I could put a hint out to the universe and the dog breeders of the world, gentle up those heads. They're a bit firm. Cool. I will link this paper in the show notes. I'm also going to link your Coastal Salish Woolly Dogs paper in case people are
Okay.
excited about that as well in the show notes. Is there anywhere else you'd like people to follow you in your work?
Um, I I mean, Google Scholar is a good.
Yeah.
I have a link tree that I can link that I can send you that has some headlines and also links for the public like articles that had and podcast episodes that had been written up on my work that like if you're curious you can check them
Yeah,
out.
that would be awesome. I think people are going to be really excited. So many dog lovers in Colorado. It's I think this isn't true. It's not a real statistic. It's a anecdote, but I believe that there are more dogs than children at this point, and it's normal to have dogs instead of children. So
Yeah.
awesome. Well, thank you so much. I have had such a good time with this interview. I'm so excited to go tell my dogs all the things I learned. I'm sure they won't care, but I do. And I'm excited to see your work coming out of University of Vienna.
Thank you. And I'm sure your dogs will enjoy the attention nonetheless.
That is true. How good was that? All right, I absolutely love this conversation. It really bummed me out to have to cut off, right? I could have gone for hours, but of course, we all need to get on with our days. So thanks for hanging out with us. I would say for me, my learn and move my feet that I'd encourage you to consider, which again, if you're new to the podcast, just one quick takeaway and perhaps thing to implement or think about throughout your week. I know podcasts can be really info packed, right? And we love to throw 10, 15, 20 things at you. And of course, you're only going to remember two of them. So I try to identify for me what was something that really stood out. So my learn and move your feet for this week that I'm going to encourage you guys to also think about is that I really loved Audrey's framework around thinking about who is your science or engineering superhero, right? Who inspired you to get into the field in the first place, especially if you're feeling a little meh right now about what you're doing or you need a little bit of an extra jazzing up. And also, if you want to expand on this even just a little bit more, is there someone who is alive and a real person, right? Your science superhero can be someone not real. As Audrey pointed out, hers was, X-Files, right? But if there's someone who is real, who inspired you that you could reach out to just to say thank you real fast, right? This might be hard. My high school chemistry teacher is one of these people, and I've tried to look him up and I cannot find his contact information, so unfortunately I can't reach out to him, but I've reached out to some of my undergraduate professors who've really inspired me in the past just to say thank you. I think these things, you don't know how they're going to land, but chances are they're going to land so much harder than you think they will, right? That person that you reached out to is really going to feel great that they heard from you and that you said, thank you so much. Song of the week. I also do a song of the week every episode. If you make it to the end of the episode, we talked about dogs, we talked about wolves, so of course I have to choose a song that has to do with either dogs or wolves. So we're gonna go with Dog Days Are Over by Florence and the Machine. Super fun song. You might know it, you might not. Give a listen. And in general, thank you for listening. A quick reminder, this podcast is not affiliated with my research and teaching roles at Colorado State University, but it's a part of my belief that we can all have jobs that we love. We do amazing things. We do things that have the potential to change lives, to change the world around us. And in order to be able to do that, we have to be able to feel lit up and alive and creative and free, not bogged down with things that carry us under our potential, right? And if you feel like you could use a little bit more help one-on-one on how do I come into my full potential, I would love to be in your corner. If you feel like you'd like to have a chat with me, please reach out. There's a link in the show notes to just schedule a quick 30-minute, totally free call where we can chat about what you find is potentially blocking you and what are some strategies to get through this. If you found this episode helpful, please share it with friends, talk about this with other people. I'm not out there doing a lot of SEO, search engine optimization. I'm not advertising heavily. Word of mouth is truly one of the ways of getting this podcast out there, and I really appreciate each and every reach out. You can follow on Instagram, STEMdentity. The music is by XSS, also on Instagram. Plenty of links in the show notes. Check it out. I love these Journal Club episodes. They're such a delight to me, so thanks for hanging out and listening to them, and we will talk soon.